Is it visible?
Yeah, sir.
Can you hear?
Good afternoon.
Good afternoon.
Today, our esteemed guest is Dr. Janmay Pahai, Senior Scientist, ICR. Sir is going to deliver lecture on eDNA technology and its application in fisheries. So you are audible, your presentation is visible. Please continue.
Thank you.
Yes, sir, please continue.
Yeah. I will be presenting you on eDNA technology and its application in fisheries. I'm working in SIPA Bhubaneswar. So, what is eDNA? eDNA is an environmental DNA defined as a genetic material obtained from water sample. This is not distinguished science of any source of any microorganisms. So this method utilizes DNA, which is excreted from the organisms in form of either glucose, gametes, or feces, blood, or other cells, not as a whole organism. And it is excreted to the environment. Okay? They say water or soil and sediment, and from that we take out the DNA. Usually, DNA we can isolate from any living organisms, it is related to plant or animal or fish. But in this case, we isolate the DNA without knowing the source. And from that, we will try to find out what was the individuals or living organisms present. So that is called, the sample is collected from environment. That's what it is called, the environment DNA. So depending on the sample size, you can see the eDNA. Here it is mentioned different nanometer size range. Here you see the multicellular species. So either it is pre-DNA or a particle bound DNA or organelle or cells or gamete tissue. They are part of the eDNA, but the size is very less. So when the size is very less, we use the sediment. And if it is bigger, from filter water or from plankton net, we can collect this. So these are the ways we can collect. Coming to the history of the eDNA, it started in 1987 when the first time DNA was isolated from the sediment. Now it has crossed many stages, and we in 2020-21, now, we have many achievements in the field of eDNA. We have developed many technologies which has been developed, has been clubbed into this eDNA to get a final better result. It may be sequencing, it may be the bioinformatics analysis. We are able to, we have advanced technologies, so we are able to identify more and more, and we are able to look deep into the system using this eDNA technology. So how we will collect the environment DNA? As I said, we collect it from the environment, either from a water source or sediment. So these are some of the sampling systems by which we collect the DNA from the water. It may be a small water body. It may be a ocean. It may be some very slow, deep water, or some any part. Even we can collect it from air also. Air from eDNA also we collect. There are sediments and water body. So different methods, these are the all instruments to collect the sample from the environment. In simple, if you see, basically in water we use these four types of them: CTD, rosette type, or ROV, AUV, autonomous samplers. What happens in this? In the first cases, we use the skin bottle. These bottles are open and they will be closed inside. When triggered to close, in some, this is the opening and closing differences are there. In autonomous underwater vehicle, what happens? The sample bottle goes inside the water, and at a particular interval, it collects the water and from that it will water it will take out like without gulping water, it will sip water, so that DNA is collected. Like this, in a autonomous sampler also, we fix that at the bottom at a regular interval. It can collect the sample. After that, we can collect the bottle or the instrument which we will use later on for isolation of the DNA. So, in simple manner way, can tell the right side it is one bottle is there. The main objective is that we have to collect the sample of water inside the bottle from a particular place, particular depth, particular time. So if you either we do it manually or through instrument, or like we have a you can unmanned instrument now, like your drones in the air. We are able to put the foods inside the water bodies to collect something. Or anyway, we have to collect the water sample of especially from when we are talking about fisheries, to collect the water sample at a different point. These photographs are maybe showing you marine photographs, because most of the works are in marine. Even we can work on the inland water bodies. Presently, we have a project on working on a like where we are collecting sample from different locations using AU. So after the sample is collected, next is target is our from that sample what we are going to get it. So it may be the objective is to identify the what is there. It may be so our interest can be to identify a particular single species or sometimes a whole community. So depending on this, we will decide or we will plan our activities. So coming to the methods. But after getting the samples, next is we will go for getting the DNA, and after DNA, what we will do? That one I want to show. So this what type of information we want? If it is quantification or identification. Quantification means we want to know how many of these species is in the sample. So for that we can use the quantitative PCR. In that we can take the sample and we can see the number of copies. How many copies of a particular, that is a very specific target species we want to know. For example, in the case we want to know particular species, what is the abundance. So in that case, we will have to take the qPCR, quantitative PCR, so that we can quantify how many copies of the particular DNA is present. The other one is identification. Identification means what are they? So in that identification can be single species or multi-species. Single species, in that case, we want to know whether this X species or Y species is present in a particular region or not. So in that case, a direct approach is we can go through barcoding, DNA barcoding. DNA barcoding, as you know, is a specific DNA fragment sequence, which we assume it is duly matched to the species level. And a particular species will have a particular DNA barcoding. And based on that DNA barcoding on DNA sequence, we can identify what is the species and or if that species is present or not. When we are looking in this eDNA, basically we look into the multi-species. Means from the sediment or environment we collect the sample, we assume in actual it happens that a lot of species will be available there. So from that we have to find out if you want to interested on biodiversity, means how many species are available, what are the species, what type of species are there. In for that we have to undergo the DNA meta barcoding. Meta barcoding, we can say a group of we do the sequencing for a and identify a group of species. Number of species we can identify in this manner. The other thing is gene pathways. Gene pathways means they really want to know the function of the storage, because in a ecosystem there are group of living organisms will be there. So they will have a interaction. Somebody um, means one how this food web is there. What are the herbivorous and carnivorous species are there, or the specimens are there, and how they are related functionally. So for that, we have to go genomics study or metagenomics study, where we have to study the functions. We have to study the different genes also, how they are present. So in eDNA, basically we will go either through barcoding. When we have a very known species, we identify. Like if it is a small location, we have a targeted, we know there are 25, 30 species are available. We want to know the evidence, how many are there, if they present or not. In that case, we can go easily for barcoding. If you have a very specific and very few number of species which you want to identify. But when we are looking into larger aspect, so here you have to do the metabarcoding. That means it is here it will allow researchers to identify many organisms against once. In one DNA sample, we will identify many species. Many barcodes will be developed. Many barcodes will be developed after sequencing. So from that sequence, we can identify or find out what are the species available in that region. So using this technique also, scientists are now trying to find out the reference laboratory. They are trying to correlate with the extinct species also. Some examples I will show you, not in this. Some examples I will show you later. So this also they are using this genomic data taken from eDNA. So if you in an overview, you want to see this eDNA, which is not the exact organism, rather it is the cell which are shaded by the feces or animals, or hair, or faces, or urine, whatever. It is coming from the different organs, maybe in a eukaryotic organism, fish, or insect, or bacteria, or virus, whatever. So this genetic materials in which are present in sediment or water, so that can be collected in terms of your sample for eDNA environment. It is actually sample, and from that, if it is a water, it has to be filtered. Then from the filter, we have to extract the DNA for proper filtration. So once DNA is available, it is ready for us to go for eDNA barcoding or metabarcoding. So as I told you, DNA barcoding will go for targeted species, target detection, species-specific primer will be used, or standard or qPCR, standard qPCR, normal PCR also we can go. And traditional sequencing we do. Traditional sequencing Sanger and all to identify the sequence. Now coming to metabarcoding, as I told you, it is a screen whole community. This we use a conjugate set of primers to identify a group of species. Then we can do PCR or next generation sequencing. Okay. So these are the basic differences between once we get the DNA, how to go ahead, like barcoding or metabarcoding. So metabarcoding, as I told you, is a barcoding of DNA in a manner that allows for the simultaneous identification of many texts within the science sample. It is a DNA-based method for identifying multiple species from an environmental sample. Combines DNA barcoding with high throughput sequencing, which is next semester sequencing. So use universal primer to amplify marker genes. So what are the marker genes? When we talk about the universal primers, let's say like animals, especially fish and all, we have CO1 marker. What is the utility of this. This is a marker. This is a sequence is fixed in that sequence. Or in that primer, all the species will be amplified. For the sequence for one species will not match with the other species. So based on this, we identify. So it becomes a marker, and to differentiate between the species to species. But this CO1 marker doesn't work into it. So for a fungi, we use ITS marker, and bacteria 16S RR marker. For a plant, we use MATK or RBC. So these are the primers we have to use for while we are going from meta marker. So in that primer, as it is universal, all the animals using the CO1 primer will be amplified, sequences will be amplified, and from that we will be able to separate out the species. So in detail, how we go ahead once we get the water sample, passes through very fine filter. This is clean and this filter then run in that filter cellulose membrane you can use basically, and that water has to pass through this filter. It's a eDNA to preserve proper this cellulose. Let's store it or use it later on, or you can use it without storing also. You can take out use the filter. So these samples has to be, I have marked proper identification, like where it has been collected, what is the latitude, what is the longitude, depth, salinity, etc. Once preserved, the eDNA samples are preserved for the lab, and we go for isolating DNA. Then we can removing the DNA from the filter. As water passes through the filter, the DNA gets attached. The DNA is eDNA, especially DNA and other things will be attached to the membrane. From that membrane, we have to remove the part. You have to isolate the DNA from that. So the sequence then after we go for sequencing, depending on we want whether we are interested for barcoding or meta barcoding. So accordingly, we will go for sequencing. If it is a barcoding, we will simply do PCR and go for cellular sequencing. But if it is a meta barcoding, we will have to go for NGS. The sequence will tell us what are the genetic information of the available, and from that we will be able to find out what are the species. So in a tabular form, if you see, this is sample collection at the soil, whatever phases, any environmental sample from where collect the DNA. This is sample is collected from the if it is water, the filter, then from filter we take out, remove the DNA, and from the DNA take after extraction, we go for PCR, then through high throughput sequencing, that is next generation sequencing we do. Then we do analysis, bioinformatic analysis, as it becomes the data becomes used. We using different bioinformatic analysis, therefore segregate the species, then the species identification and follows. After that, we will do analysis based on our hypothesis. And so steps that can be like a collection of environmental sample, second DNA extraction, amplification with DNA markers, which is CO1 and all I have discussed. With this marker, we amplify and we go for high throughput sequencing because next time sequencing, then bioinformatic analysis, species identification, next is analysis. So what can eDNA tell us? Am I audible? Hello? Anyone, please? Hello? Hello? Am I audible? Hello? Can you hear me? Anyone please respond?
Sir, please share your PPT.
Okay. Yeah, now it is audible.
Okay, sir, please continue. Yes, sir. Yes, sir.
So now coming to what can the eDNA tell us? So the eDNA can tell us what are the species present in a particular location that can be used to assess the biodiversity, monitor the species changes over time. Second one is but it can say also how many species present in this. What it means, it is abundance. It also can say the movement or habitat associated with endangered rare species or migratory species or any cryptic induced species, whether if any available there in that region at a particular time. Then also water quality, like detecting invasive species, harmful algal blooms, pathogens, parasites. So these all water quality related things it also can tell us. Another most important thing which is not also scientifically proved, because in some time it is associated, in some cases it is not, links between the eDNA and any other properties which link us famous for the species distribution. So for this is not a direct we can eDNA can tell. We have further analysis or other data to interpret to get this. So these are possibly eDNA we can get information from. But what we cannot get it from eDNA, we may think that we will get it, but it is not. Like eDNA cannot say a sample is from a living one or dead, whether it is from a resident species or migratory species. But for that reason, if I know these are the resident species of a particular location, and if I find somebody else, some other species, that we can say it is a migratory or not. If we have no idea, it is very difficult from the DNA to tell whether it is a resident species or migratory. So exact number of species or individual, exactly cannot say. You can say abundance. Yeah, it is present in more or less. You cannot say that 125 numbers of a particular species is found in this region. No, it is not possible. Then again, one more is the size, age, the life stage of the species, which is that where the sample is cannot tell what is the size of the fish or age of the fish, or whether it is at or larval stage. The fish we cannot tell. And the absence of a species that cannot be also taken. It may be from the sample we have collected that particular species had nothing in this environment, so we are not able to know. So we cannot exclude that a particular species is not available in that case. Okay, so overall it can tell us what are the species available at a particular time. So if you want to use the eDNA technology, this is so I have noted applications like fish biodiversity monitoring. eDNA will tell you the number of fish species, diversity, it will distribute across aquatic ecosystem. It helps to find out the cryptic, rare species and provides the real-time position of the number of the species available. So it can help you monitoring the particular biodiversity, and so it also suggests you whether any type of disturbances in a particular biodiversity may be because of your climate or because of any human interference like a dam and all. Any changes are occurring in the course of time that you can have an idea. Then early detection of invasive species. Invasive species are not wanted at all. So this invasive species can be detected in not in the later stage. We can detect in the early stage, so that we can take notification. So this allows sensitive and early identification of invasive species by detecting their genetic material in water sample. When they are less in number, also you can, if you are having the sample, you can detect before physically you see that. Then is pathogen surveillance. And like a bacteria, virus, fungi, these pathogens, we see when there is any infection, there is any disease prevalence. But before that, if you can identify this pathogens, we can prevent the disease and take notification of it. Eutrophication or algal bloom control. The detection of algal DNA. Again, help to monitor and manage the eutrophication and the harmful algal blooms. We get early warnings. We can main thing which is that we can identify the algal DNA, which can be expected to have a bloom fusion. So if you identify these are the species of algae is found which can make a bloom, and you can accordingly you can act before. Next is the water quality assessment. In this, you will have the composition of microbes, plankton communities, other bioindicators, which dictates, which says about your water quality. How many, what are the beneficial microbes are there, or what are the planktons present which are required of this, or to also know that also help you identifying a detection of pollutions. Many bioindicator species are there for pollution, so that it can be easily identified. Then identification of the larvae and the egg, especially in our project, we wanted to know how this is species is breeding, when it is breeding, when it is, what is its there is a breeding ground actually, because there was human interferences, so the water was sinking, and which was leading to the decrease in population. So we are trying to find out the eDNA from based on eDNA information at which location we are getting the larvae, and when when means this breeding season is maybe 4 or 5 months, but at a particular time when it is breeding, later you can get you are getting the this larvae at different locations of a particular targeted region. It is very difficult in manually to know what are the larvae and fish where it is available. If you collect the samples regularly from different locations and have a and in our case, it was very specific, so that we can go for perfect. Because we were interested for a few 4 or 5 species, we wanted to know where they are available, and whether their evidence is going up and down. That's what we used barcoding approach. But other way you can go for meta barcoding when we are looking into a larger aspect. Then monitor antibiotic resistance genes are also you can identify from the eDNA. So antibiotic resistance gene is present in the bacteria, you can identify from the eDNA. These are all we discussed about the fishes, some examples. And to make it something more, and let us have some information from outside the fisheries. This wildlife organization, they have this eDNA program for the conservations. There are 4 or 5 examples. This first one is searching for rare aquatic and terrestrial species in water. What does it mean? These very rare species which were not able to they're not able to find, they are able to try to get it from water. How, and is another problem that there are still species in water. What is this meaning? Meaning is that when this species are in the, let's say, in jungle, somewhere there, so they used to set their species which goes by water to the water, flows with the water, rains. So from the rain or a water source, you can collect, you can see that your eDNA. From there, you can identify, yes, so here this type, this species, these are the species are available. You cannot go physically inside to deep jungle to find out whether they are there or not. So this helps in monitoring the wildlife. And further, one is that monitoring or identifying individual polar bear from the eDNA in the track. Whatever, I have one more slide. I will say all this polar bear also how they do. And the last one is your counting tiger prey in water. So what they the objective was, they wanted to protect the tiger. So if the tiger, one tiger they require nearly 4 to 5 kgs of meat every day. Accordingly, they must have the prey, like the deer. This much of a deer should be available. So they want to know how much deer is available. Get the information on deer. They use this technology to identify how many deer are present, so that these tigers are safe. It is a one type of conservation mechanism for which they are using this eDNA from the eDNA technology for this larger scale. Identifying, as I told you, the polar bears, what happens? So when the polar bear walks, the eDNAs are left behind as paw prints, or any polar bear or any animal when it moves. So this it is polar bear is in the snow. If it is not in the snow, it is in case of it can be in a terrestrial jungle with the animal. So the paw prints or the whatever the said DNA from that animal gets washed by the water or it is snow. Here in polar bear, they have collected the snow, and they have, from the snow they have, when it melted, they capture the liquid passed through a filter to capture the DNA. The DNA sample was extracted to use identify the what type, which polar bear was there, how many polar bears are there, etc. So this was the they have used this eDNA technology for identifying individually polar bears from the eDNA in their tracks. This is one more important use, that is ancient environmental genomics. What it is this? They reconstruct past environments using the ancient eDNA. So ancient eDNA is like eDNA is like if you have a fossil, from the fossil you can isolate the DNA. From the DNA you can try the whole genome also, and you can study. But where there is no fossils, in that case there are possibility that there are some cells or some hair or some pieces, something, if it is there in any particular layer of soil, and they take out this, that they can use for the understanding the changes in the environment. What it does mean is this, as you know, the particular this evolution is happening, the environment is changing. So at a particular time, this environment has changed. So prior to this, what are the genetic materials? After this, what are the genetic materials? So what are the adaptation it has taken from the genomic data, which is we don't have. They are trying to, this group is trying to get it from the eDNA. If you have a if you have the DNA, that is okay. But when you don't have, so to reconstruct the genomic data, they you can use the eDNA also. eDNA means there are body parts. There's some parts they are places like one. From there we have to take out the DNA, and it is very difficult to do. You may not do it because the DNA is a life. It can survive up to 1 million years or so. But that will be after, as you know, DNA will start degrading maybe for 500 years, the half-life in some of the studies it says. But it can, if somehow it is protected, it can be there up to 1 million years. So in that case, if we find some DNA, part of DNA, 1,000 years ago or so, which has been extinct and there is no fossils, so that we can, that they are trying to use. So they have published some article and all. And so in fisheries, there's some examples like anemone in Japan. I've taken this slides from. So this anemone, they have been using the eDNA technology. They are trying to identify different locations, 860 locations or something. So from there, they are collecting the information and species available, so that they will have a particular this species, this fauna of the whole Japan. So they can use it for their use as well as that will be for the scientific use. The whole information will be available, but this information usually we collect the case, we identify the case, then we catalog this, then we find out this the pieces, what they are engineered from the eDNA. So one slide I have taken for a plant. So in that they say many in case of a plant, all are environmental DNA. All are environmental DNA basically from the natural system. As a agricultural plant system is fully on natural system. So they have all natural like airborne or freshwater marine DNA, soil microbes, wasted pathogen, all are part of this natural things, and as well as plant ribosomes. Also, they hold together. They are used for plant science using this eDNA technology. So this you can use this environmental DNA technology like this in case of agricultural science. So one more thing I have found somewhere, it is subway air microbes. Means in case of subways enclosed metro system, we close the system. So what happens? The bacteria, fungi, they are present. They are air. So from air also detection is possible. eDNA. From there we can detect other bacteria and viruses. This mechanism is a little bit different. They have to like collect the eDNA. They have to collect from the air and they are collected from the water. It is different. So the filtration mechanism is a little bit different. So the point is this airborne eDNA also it has a second role, but it is a role in case of our health. So what are the challenges? Challenges, as it has many benefits, there are also many challenges. Like while collecting the sample, there is possibility that we are not perfectly collecting the sample, and again the sample collection, there is chances of contaminations, and the detection method may be not proper. Then there will be usually appliance by taxonomic like on a signed sequence that is as well. Many of the species who do not have the previously reported sequences that we cannot identify. So incompleteness of genetic reference database, that is whether if we have a reference database, then we can say it is present or not. Like we have to know the originally the specimen. After that we can check. Then some very rare elusive species for misidentification also. eDNA, the spatial and as well as temporal dynamics, there will be challenges because a particular type you cannot say whether a particular species is present here or how far it is from. Or it is, you cannot say it was present today after 1 hour, before 1 hour or before 10 hours. Sometimes it becomes difficult to know. And inconsistency data analysis and interpretation system, which is not the same. So in conclusion, I can say there is a great potential to extend the biodiversity assessment and exploration, although it needs standardization protocols like research, sequencing, data acquisition, analysis, interpretation. Along with we need the best as a reference data. So importance of eDNA data mobilization and integrated eDNA database that is there. It's findable, accessible, interpretable, and reusable data, so it can be used for open science. At the end, I can say it has enormous power and potential of eDNA. It can revolutionize the fisheries and aquaculture by precise, efficient, and non-invasive monitoring of species, ecosystem, and water quality, mainly on biodiversity and ecosystem management. For detecting fish diversity pathogens, to tracking harmful bones, invasive species, and antibiotic resistance. eDNA offers a multi-purpose platform for our ecosystem. Further, by supporting early detection, real-time monitoring, and from some eDNA contributes sustainable aquaculture management. One big improvement is DNA sequencing technology and bioinformatics can help us expand a more scope and more efficiently, more sensitive to eDNA application. So, you can say eDNA technology marks a paradigm shift in aquatic monitoring, fostering underway, enhancing biosecurity, and ensuring ecosystem resilience for future generations. Next one. Thank you. I think I finished.
Hello.
Hello.
Thank you, sir.
Yes.
Thank you, sir. Now participants can ask questions.
Welcome.
Sir, I think there is no question. Once again, I would like to thank Dr. Janmay Parhi for this wonderful session. Thank you, everyone.
Thank you. Thank you.
So now the session has come to an end.
Thank you, madam. I will leave now. Thank you.
Thank you.